Okay, everybody, we are down to our last chapter, chapter 18, translation. So protein biosynthesis. So if we think about before what we've talked up to this point, so we talked about DNA and DNA replication, right? So the DNA needs to replicate. It's what's called semi-conservative, meaning that the original strand is going to stay. It's going to split. New strands will form, and then those strands will split. So like the parent DNA is always still going to be present in the new is going to be present and passed down to the new cells. Then we talked about transcription. Transcription is the process of actually creating mRNA. We talked about how we're going to have the RNA polymerase. It's going to be running along the DNA. It binds to a promoter region. Within that promoter region, we're talking about the TATA, the TATA boxes. That helps the RNA polymerase to bind to the DNA. When it binds, it's going to go ahead and transcribe the gene, the chunk of information that we want, all the way until it reaches a quadruple A in the DNA, which creates a hairpin loop. So it's going to have, at the very end, it's going to U, 4 U's it's going to put down, which creates a hairpin loop and helps pop off the mRNA. There's also a Rho factor, which is another protein which helps to terminate the RNA polymerase function and help pop it off of the DNA. Now, we don't use this entire mRNA strand. There are introns, which are non-coding parts, and then exons, which are the parts that we actually use. Those are the coding regions. So we're going to have a spliceosome. The spliceosome is going to split up the chunks, the exons and the introns, and we're going to take the different exons and splice them all together in order to create a final product, mRNA. And we also have, there's going to be a cap that is placed on the 5' end. And then there's also going to be a poly A tail, so a whole bunch of adenosine at the very end is going to be put there at the end of the RNA on the 3' end in order to protect that mRNA strand from nucleases that would want to break it down. So finally we get to that point, we have that completed mRNA. All of that has taken place in the inside of the nucleus, and now we're going outside of the nucleus. The mRNA is going to be transported through the little pores in the nucleus wall, and now we're going to get to translation, which is the next step, actually getting that protein built. So objectives, we're going to describe the genetic code, we're going to describe the effects of mutations, and also describe the process of translation. So we talked a little bit about mutations before. That was in chapter 16, so if you don't remember much about the mutations, jump back over to chapter 16 and just read about a couple of those really quick. Okay, so protein synthesis or translation of mRNA. So translation is a process by which mRNA ribonucleotide coding sequences are decoded. So the mRNA ribonucleotide coding sequence, that's just the mRNA that we made from the DNA, are decoded and directs biosynthesis of polypeptides or proteins through peptide bond formation. So hopefully this is sounding a little bit familiar from cells and tissues. Polypeptides is a chain of amino acids. It's a chain of amino acids, and those will eventually get folded and form functional proteins, and they combined together through peptide bonds. We'll also talk about peptide bonds being they're also covalent bonds. Okay, so this peptide bond formation followed by its post-translational processing and modification. Okay, so the genetic code. So the nucleotide sequence that is present in the mRNA contains a code for each amino acid. This is known as a genetic code. So there's a code for each amino acid. We'll talk about this down here a little bit more with the codons. So one gene may contain more than one codon specific to a protein or a polypeptide subunit. Cistron is the smallest genetic unit that codes for a single peptide. Although previously it was known that one gene expresses one protein, the concept can be best described as one gene expresses one polypeptide, or one cistron can express one polypeptide. All just saying the same thing. Just know that there's one gene to one specific protein. They're linked. You can't get a different protein off of the same gene. It's always going to code for the exact same protein, and if you create the same polypeptide chain, it's going to fold the same, and you're going to get the same functional protein. So one gene to one protein, and one protein to one gene. So this concept arose because many proteins contain subunits for the formation of their quaternary structure. We talked about quaternary structure, multiple different polypeptide chains. After they've been folded in their tertiary structure form, they come together to form an even bigger protein altogether. Genetic code is a specific sequential arrangement of codons that specify all the amino acids found in proteins. Specific 3 nucleotide sequences of mRNA of the mRNA molecule codes for specific amino acids. So 3 nucleotides. These 3 nucleotide sequence is known as a codon. So codon, a sequence of 3 bases, or also called a triplet, in the mRNA 5' to 3'. And this codon is specific for an amino acid. During translation, successive codons in an mRNA determine the sequence of amino acids in the polypeptide. I'll go ahead and show you this ahead. These are all of our different triplets that we can have for amino acids, and connecting them what they are to each amino acid. So you can see that some of them code for the same, like AUU, AUC, and AUA, all code for the same amino acid. So there can be duplicates. And we'll get into why that's important a little bit later on when we touch more on mutations. So there are 20 amino acids required for the synthesis of proteins in humans. 61 codons represent 20 amino acids, plus 3 codons represent stop codons. So there are a total of 64 codons present. Also note the triplet sequence is written in 5' to 3' and direction. The third base of each codon is less important in specifying an amino acid than the first 2 bases. The reduced stringency between the third base of the codon in mRNA and the complementary anticodon in tRNA is referred to as wobble. Real quick, anticodon in the tRNA is the transfer RNA. The transfer RNA is what is going to carry the individual amino acids, and the anticodon is what matches up onto the mRNA, matches up onto the codon to then bring that amino acid to allow that amino acid chain to form. So kind of this little bit of a wiggle room, especially towards like the third base of the codon, is referred to as wobble. So due to presence of this wobble position, one amino acid can be represented by more than one codon, and we'll see the chart below. So the genetic code is degenerate. So redundant. Each 20 amino acids has at least one codon. Some amino acids have multiple codons. This is one of the interesting characteristics of the genetic code. A specific amino acid can be coded by more than one codon. All the amino acids except methionine, this is going to be important. Methionine is our start codon, AUG, which codes for the first amino acid, methionine, added during protein synthesis. So methionine is our start codon. Every single one of our proteins is going to start with AUG. And it's going to, the first amino acid is methionine. Except methionine. And tryptophan, represented by the codon UGG, can be represented by more than one codon. Hence the code is degenerate. big one to remember here is methionine. That's our most important one. Genetic code is also unambiguous. This means a specific codon only represents one single and specific amino acid. Hence, the genetic codes are both unambiguous and degenerate. 3 codons that do not code for specific amino acids are known as nonsense, or stop codons. UAA, UAG, and UGA. If one of these codons pops up, the process of building the polypeptide ends and it stops and everything releases. So which is why when we get certain mutations, that can be an issue because it can mutate to a stop codon, and then that's an issue because then that protein cannot be formed. Because every time it tries to, rather than putting an amino acid there, it just stops the whole process partway through. So presence of any one of these stop codons at the 3' end of the mRNA transcript causes termination of further peptide bond formation during protein synthesis. Genetic codes are non-overlapping. During translation, these non-overlapping triplet codes are read successively, one after the other. So we can see here our beautiful little chart of all of our different codons. Big one to remember, AUG. I'd also probably recommend knowing the stop codons. It's probably a good idea as well. So the genetic code begins with AUG near the 5' end of the mRNA, and AUG is our start codon. And then we have our stop codons right there. So what this means is, like, if we look at our codon, so let's say we have a AUG, GUU, GUA, GCA, GAA, something like this. That's our strand of mRNA. So, and actually, we could even actually have it look like GGUA, CGUAC, something like that. Looking through this, you could have a question like this: which of the following, like, what is the proper string of amino acids based on this chain? And you're looking at this, everything before the AUG codon is trash. It's garbage. Because it doesn't matter. Because you need a start codon in order for this whole process to start. And then from there, you just divide it by triplets. So AUG, GUU, GUA, GCA, GAA. And so we have methionine. GUU is valine. GUA is another valine. GCA is alanine, and GAA is glutamate, or glutamine. And that's exactly how that would be written, where then this is now our polypeptide chain. This is the polypeptide chain that would be formed from this specific RNA sequence. And you can actually even take this backwards and ask the question such as, what would the DNA sequence be for the following polypeptide chain? And it gives you this polypeptide chain. In this case, if you were to ask this question like this, he has to provide this chart, or you have to be able to have access to this chart in some form or another, because otherwise, to be honest, memorizing every single one of these is a little bit difficult. But you could look at this, like AUG is on the RNA, so then what would that be looking like on the DNA? So in the DNA, A goes to T, U goes to C, and G goes to, U is T, so U goes to A, and G goes to C, because it's at the golf course, how those pair. So you could start to write the DNA code all the way back. I don't recall, I feel like I recall him asking one question or so like that. Nothing too crazy. But yeah, again, you should have this little chart in order to make that actually doable. Okay, so talking about mutations. So mutation it is a change in the nucleotide base sequence of DNA. A mutation can be due to replacement of one or more base pairs with another. That's called a substitution mutation. Or due to addition or deletion of one or more base pairs. Addition is an insertion. Deletion is just called deletion. So this is something like UAU, that is a tyrosine codon. Let's say that it gets converted, there's a replacement where a G replaces the U, and all of a sudden it's a stop codon, and that protein can't be created. Or you can look at maybe the A goes to a U and you get a new protein. Or sometimes you can have what's called a silent mutation, where yes, it mutates, so this U goes to a C, but because that codes for the exact same amino acid, nothing happens. And those silent mutations where it's mutated, yes, but it really doesn't matter. Occasionally as well, you can have it if it mutates to an amino acid that holds very similar properties. You can still get partial function of that protein. One example of that is sickle cell anemia. So with sickle cell, if I remember correctly, I don't know if he talks about it in this chapter, but we talked about it way long ago. I think we'll get to it. If I remember correctly, it is a single valine is switched or replaced or something like that. There's a single mutation in that sickle cell trait, which causes the hemoglobin to really not function, for the whole structure of the red blood cell to be poor. The whole red blood cell itself doesn't form quite properly. So you can see the power that these mutations can have. So going through a couple of these, so point mutation. A point mutation results due to replacement of one base with another. Replacement of one base with another. A purine base or a pyrimidine base can be changed to another, or one purine base also can be changed to pyrimidine, or vice versa. And this can result, so a point mutation can result in one of these three. The silent mutation that we talked about, where it codes for the same one. A missense mutation, which then it codes for a different one. Let's read this way. So it can happen due to replacement of a single base in the codon, specifying a different amino acid other than the actual amino acid represented by that original codon. This way, a missense mutation can produce variable phenotypic effects. So you can have changes in that phenotype. If I remember correctly, a missense mutation is actually what sickle cell is. Nonsense mutation. This type of mutation can be produced when a stop codon is generated due to a single base change. It can terminate the protein synthesis immaturely or can form a non-functional protein molecule. So you can see what these happens. All three of those are examples of point mutations. We can also have insertion or deletion mutations, or in this case, one or more nucleotide are inserted or deleted from the nucleotide sequence, and we get frame shift mutations. So this type of mutation occurs due to insertion or deletion of one or more nucleotides not divisible by 3, making a shift in the reading frame or grouping of the nucleotide sequence. So kind of like here, like how are you, we slice out the W, and now it goes to HOA, Ray, O, and then this doesn't have a codon, and now we're coding for completely different amino acids. So it would have completely different codons and anticodons. The code, make sure I'll start right here. The code is a triplet code formed by 3 nucleotides that specify an amino acid. A change in one base, either by insertion or deletion, is able to change the entire sequence of amino acids in a wrong way, forming a defective or incomplete protein or polypeptide. One illustration can be given below, this whole example that we have. The next example is an insertion. If we insert one, so this is a deletion, we can also have an insertion again, which shifts the frame, and that completely changes. Or you can even think, let's say if we have like a start codon for methionine. And then all of a sudden we inserted another A here. That protein can't even start because it doesn't have the methionine start codon. And maybe it has it a little bit later on down the road. Yeah, I could have it a little bit later down the road, and then suddenly the protein is, or the polypeptide chain is starting at the completely wrong point in the gene. So here are the examples of the insertion and deletion. And you can see here kind of how this changes. Inserting a C in the third triplet, inserting one here, we suddenly get a whole different form. Deletion, we get different amino acids. So these are our insertion or deletion mutation. Pairing relationship of the codon and the anticodon. The specific codon in mRNA is recognized by the anticodon sequence that is in the specific transfer RNA, or the tRNA. The fundamental basis of recognition of codon-anticodon sequences in mRNA and tRNA, respectively, is followed by complementary base pairings. So very similar exactly to how DNA-RNA works, or how the two different sides of DNA work. They complement their base pairs. tRNA to mRNA is the exact same thing. tRNA are crucial structural RNAs with some functional basis for the translation of mRNA into respective amino acid sequences, resulting in a specific polypeptide. However, tRNAs contain some unusual bases, unlike mRNAs and rRNAs. Some of these bases are methylated, deaminated, or reduced. In pseudouridine, the base uracil is removed and is attached to the sugar via C5 and produces a glycosidic linkage. tRNA also contains thymine residues, as in a T. I think that little guy is omega. Let me look it up. It's psi. That's a lowercase psi. So T psi C. I'm not sure exactly what that's supposed to represent. Thymidine pseudo. Okay, that's the symbol for pseudo. For pseudouridine is the psi, PSI, in the structure. So we have tRNA. Here is our psi arm. It creates a little hairpin loop, and that's just a little part of the building of the tRNA to have the anticodon and the codon to pair up to the codon. Sorry, give me one moment. Alexa, lamp on. Apologies about that. Okay, so the tRNA has a clover leaf-like structure. A typical tRNA contains 4 base pair stems, their hairpin loops, and 1 hinge. 3' N contains a sequence, 5' CCA, 3' OH, which binds specific amino acid. So here, this is where our amino acids are going to be bound, and this is where our anticodon is that can bind to the codon of the mRNA. And we have 1, 2, 3 technically hairpin loops, and a hinge as well. This is giving you the general structure, and this is how it's bound right here with the phosphate onto the amino acids, how it carries the amino acids. Protein synthesis. So protein biosynthesis occurs in a few steps as described below. First is activation of amino acid by amino acyl tRNA synthetase. Catalyzed reaction. We'll just start there. Go through these one by one. Okay, so activation of the amino acid by amino acetyl tRNA synthetase. So keep in mind that the enzyme substrate reactions are very specific to their substrates, and enzymes in a typical biochemical reaction. This is what we're talking about, how each enzyme is going to perform a very specific task, and it's not going to deviate from that task. So that's just reiterating that point here. Each amino acyl synthetase recognizes a particular amino acid and the tRNA specific for that amino acid. The specific amino acid is attached to its specific tRNAs at the 3' hydroxyl group by the amino acyl transfer reaction catalyzed by amino acyl synthetase, forming amino acyl tRNA. Thus, each of the 20 amino acids is attached covalently to their respective tRNAs. This process is energy dependent and requires hydrolysis of ATP. So this whole process that we're talking about is binding the amino acid onto the tRNA, and it's a very specific reaction. So each of the tRNAs are going to be specifically built to hold a specific amino acid. And this amino acyl synthetase is going to recognize that particular amino acid and the particular tRNA to pair them up and bind them together. And it's going to break an ATP and use ATP to form that phosphate bond, just like that, right there, in that process. It's a hydrolysis reaction using ATP. And it also uses magnesium as the required cofactor. Magnesium is super, super important in a lot of different pathways. A lot of people tend to be deficient in magnesium because they don't eat enough vegetables. You'll see that this is not something to encourage everybody. Obviously, you stay within your scope of practice, but there is a lot of benefit to individuals taking magnesium supplements, as it is a cofactor to a lot of different reactions in energy production and obviously protein production as well. So this covalent attachment of the amino acid to its specific tRNA keeps this molecule in its activated form. The first amino acid in the synthesis process is methionine, always methionine, which binds to the methionine tRNA at its amino acyl site by a covalent linkage and forms methionine tRNA. This methionine tRNA is formulated at the amino group of the methionine residue and is described as F-methionine tRNA. Don't worry too much about all of this extra stuff. Just understand it's like the name of the amino acid and then the tRNA because it's specific for that amino acid. Step 2, initiation of translation. So in the case of eukaryotes, including us humans, the ribosome contains 2 subunits. We describe these by something called the sedimentation velocity. Basically, they take the ribosomes, they split them into different parts, they stir them all up, and they wait to see how long it takes them to sink to the bottom. Because if something's heavier, it's going to take less time to sink, and that's how they figured this out and measured these. So the sedimentation velocity of the smaller subunit is 40s, so we call that the 40s subunit, while the sedimentation velocity of the larger subunit is 60s. The sedimentation velocity of the combined ribosomal subunits is 80s. It's not like good math. You want it to be 100, but it's not. It's 80. You will most likely get a question on that. So know that the small unit is 40s, the large unit is 60s, and combined, they're 80s. It just, again, has to do with the amount of time and everything that it takes for them to sink when they're combined and then stirred up in a solution. Okay, so the 40s subunit, the smaller of the two, contains 3 sites. It has an A site. The amino acyl tRNA binds at that specific site, at the A site. The P site, the peptidyl site where the peptide bonds are formed, and the E site, the exit site. Both A site and P site can bind amino acyl tRNAs. E site can only bind uncharged tRNAs after completion of their functions on the ribosomal sites. See if he gives us a good picture here. kind of an okay picture. We'll see if we can find a better one in a minute. So initiation begins with the binding of eukaryotic initiating factors. There's about 12 eukaryotic initiating factors, but don't worry too much about that. Just understand that it has initiating factors. And those initiating factors act on the 40S subunit. Functionally, these eukaryotic initiation factors make sure that the A site is blocked for the methionine tRNA, and only P site is available for F methionine tRNA. EIF3 binds to the E site and inhibits premature combination of the 40S and the 60S ribosomal subunits before the elongation phase begins. GTP bound EIF2 binds with F-methionine tRNA, and formulation of methionine keeps it as a charged molecule ready to participate in the reaction, and also to be very specific to bind only at the P site of the smaller ribosomal subunit 40S. It's kind of saying a lot. From this paragraph, good thing to understand, look at because it names these specifically, I'd take a look at these two and understand, just know what those two specifically do, as that'll help. You could get a question or two thrown in about those specifically. In the next step in this initiation process, the mRNA to be translated binds to the 40S subunit of the ribosome. Okay, so the mRNA binds to the 40S subunit. The 5' cap of the mRNA detects and binds to the 40S subunit. So that 5' cap not only is protecting the mRNA, but is also now going to be kind of our signaler to bind it to the 40S subunit. The start codon, or initiation codon, AUG, is placed at the P site. The detection of the AUG codon, its correct placing and binding to the P site, is guided by a sequence known as Shine-Delgarno sequence. That is localized at about 8 to 13 base pair to the 5' side of the start or initiating codon. The next step is followed by binding of the F-methionine tRNA eukaryotic initiation factor 2 GTP complex. It's just saying all of that stuff that was together is all just kind of bound together, and now it's called a complex because it's all stuck together in one spot. Specifically, so it's all bound specifically to the P site via the anticodon, UAC. So AUG is the codon, UAC is the anticodon. And so it's bound to that anticodon recognition arm, which matches the base pairs with the mRNA, AUG codon, in the EIF2 and GTP dependent manner. GTP is hydrolyzed, powering the process, and the EIFs are released free. At this point, so this is up to this whole point, we have been getting the mRNA. The mRNA is now bound to the small subunit of the ribosome, the 40S subunit. And our very first methionine has come in and locked into place, where we're going to start. So this is where we've gotten up to right now. So we found our start codon. Our very first tRNA has bound. Its anticodon is stuck together, and the GTP and all this process have popped off. Now at this point, the large ribosomal subunit, the 60S, binds and completes formation of the functional initiation complex. Thus, the functional initiation complex contains the mRNA, the amino acid tRNA, so it's just the methionine tRNA, beginning, or whatever amino acid it is containing, and the 2 ribosomal subunits. And we're here. So it's all kind of sandwiched here together. They're all in the P. There's an A spot, there's a P spot, and then there's the E, the exit spot. Now we have peptide bond formation and elongation of the peptide chain. So now we have the first step, the first amino acid, the methionine. We need to start adding amino acids to this chain to grow it to eventually be able to form our protein. So the elongation step requires mRNA, amino acyl tRNA, and 3 cytosolic elongation factors. Yeah, those are these guys listed right here. If it helps you, I would probably write out these steps because there's a lot of words here. I would write them out a little bit more concisely and just kind of where you see these specific factors, and that'll help you prep for the test. If you get any kind of tricksy questions specific about these sort of things. A proper incoming amino acyl tRNA binds to the GTP-bound EFTU, forms amino acyl tRNA EFTU complex, and guides and attaches the second amino acyl tRNA to the A site of the initiation complex. The A site, which is attached to the second codon of the mRNA, now recognizes the incoming amino acyl tRNA containing the appropriate anticodon on it. GTP hydrolysis powers the reaction and causes release of EFTU from the site. EFTU GDP complex is recycled back with the help of EFTs in a GTP-dependent manner. Literally all this is saying is that the next one is coming in and getting stuck in here in the A site. So methionine is in the P site, and the next one gets recognized. The anticodon matches the codon, and it enters into the A site thanks to some of these other factors, and it uses GTP again. It uses GTP to get it in there, and then all that breaks off, and then you have one tRNA in the P site, one tRNA in the A site, and then we're going to connect the two. Okay, next the amino acid into adjacent amino acyl tRNAs in the P site and the A site forms a peptide bond by condensation. In this condensation reaction, the first amino acid F-methionine is transferred over the new incoming amino acyl tRNA located in the A site. Thus, a dipeptide is formed on the A site, the F-methionine being the first amino acid, and the second amino acid is attached to the tRNA on the A site. This transfer of amino acid from the P site over the amino acid on the A site is catalyzed by the peptidyl transferase activity of the ribosomal RNA. Next, in the translocation step of elongation, the ribosome complex moves one codon ahead in the direction of the 3' end of the mRNA. So it shifts down 3 codons, or 3 base pairs. Or 3 base pairs, or 1 codon, 1 triplet, depending on how you want to say it. As a result, the new dipeptidyl tRNA is positioned at the P site, and the empty uncharged tRNA produced from the transfer of F-met moves to the E site, leaving the A site empty for the next third incoming amino tRNA. The third amino acyl tRNA comes, and I'm not sure what that word is supposed to be. It comes and it lands and it attaches at the A site, followed by the next peptide bond formation when the dipeptide from the P site is transferred over the third amino acid in the A site, producing a tripeptide on the A site. The ribosome moves again ahead 1 codon towards the 3' end, and the peptide chain elongation cycle continues until a terminal stop codon is reached. For each amino acid residue attached to form peptide bond in the elongation polypeptide chain, hydrolysis of 2 GTP molecules provide energy for the movement of the ribosome 1 codon in the direction of 3' end of the mRNA molecule. So it literally just kind of attaches, so this pops over, and then it slides down. So it slides, and then it pops over, slides down, and the next one comes in. Pops over, slides down, pops over, slides down, pops over, slides down, over and over and over again, and these keep on getting filtered out. Next step is the termination. The termination occurs in response to the termination codon in the A site. Real quick, there's a lot of words through all of this. Makes it sound a lot more complicated than it is. It's really not that bad, where you just have it, we get the setup and everything in the P site, the new tRNA with the next amino acid enters into the A site, it transfers over, and then it slides down as the next one comes in. The one leaves, and then it transfers again, and it slides down and slides down. It keeps sliding until it reaches the end. It's not that complicated, don't worry. The only thing that's part about this is complicated is you just need to work on then getting used to all of the vocab, the different names for all the different stuff. The different names for like the tRNA, the different chunks of the ribosome, the small and the large portion, as well as just some of the enzymes. So termination occurs in response to a termination codon in the A site. And these are our termination or stop codons. Elongation of the polypeptide chain stops when the ribosome reaches any one of the stop codons. At this point, at the stop codon, a ribosomal release factor is bound. Presence of this bound ribosomal release factor causes release and dissociation of the ribosomal cell units. So the 40 and the 60 pop away from each other. mRNA transcript and the uncharged tRNA. This reaction is mediated by hydrolysis of EFT-GGTP complex and aided by a ribosomal recycling factor, and also EIF3. The role of EIF3 is to bind with the 40S ribosomal subunit and repeat the process of initiation of translation. Here we go. We get the release factor comes and it binds, and everything breaks apart, and off that little polypeptide chain goes. Step 5. Post-translational processing and modification of the polypeptide chain is required for its proper function. We talked a little bit about this a while ago, talking about the folding and all that sort of good stuff of the polypeptide chain. So newly synthesized polypeptide chain gets its three-dimensional tertiary structure through folding and processing appropriate for its function. If more than one polypeptide chain are synthesized from the polycistronic sequences during gene expression, then they can form quaternary structures. Adjustment of the amino and carboxyl terminal. The first amino acid in the newly synthesized polypeptide is methionine. If the normal form of the polypeptide does not contain methionine as its first amino acid, it can be removed from the amino terminal. Sometimes removal of the carboxyl terminal amino acid is also required. So it can cut off the beginning or the end, is what that's saying. Just in case that protein doesn't actually use methionine, it's going to know that, and it's just going to snip it off. Loss of signal peptide. So presence of a signal sequence in a polypeptide is often important for its transportation of target cells. Therefore, the signal peptide can be removed. So if there's a specific peptide signaling for this one to be formed, if that signal is no longer being sent, then it's not going to continue to do its function, because if the signal is not being sent, it means it's no longer needed. Individual amino acids are also altered. Some amino acids can be carboxylated to provide special functions. Some proteins can form metalloproteins or attach prosthetic groups. So the phosphorylation of serine, tyrosine, and threonine, etc. Certain amino acids can be phosphorylated at their hydroxyl residues. Attachment of carbohydrate side chains. Some amino acids can be glycosylated by forming O-linked or N-linked oligosaccharides. Think about oligosaccharides, types of sugars. Addition of isoprenyl groups can occur. Some small G proteins known as ROS proteins are isoprenyl derivative and are products of oncogenes and proto-oncogenes, and thus have implication in cancer biochemistry. Onco is talking about, that's a root for cancer. Addition of prosthetic groups, metal ions to form metalloproteins. Proteolytic processing is also required to generate active forms of some enzymes or peptide hormones, such as insulin. Formation of disulfide cross-linkage is often characteristic in protein structure. We've talked about that before in the early chapters in this book. last and final examples of some inhibitors of translation and their mechanism of action. Puromycin inhibits peptide bond formation. Calramphenol and cyclohexamide inhibits the peptidyl transferase. And that, my friends, is all of Biochem 1. Good job making it through. Just have one last hurdle, the final exam. Hopefully you guys have enjoyed going through these videos. Hopefully they've been a big help to you guys. If at any point you need something, feel free to reach out, drop a comment, or send me a message if you have my contact information. And I wish you guys the best of luck on your final exams and your rest of your career here at school. Thank you and goodbye.